only 3 drugs to know from Part 1. Orlistat, which is Xenical ally, pancreatic enzymes, and ursodeoxycholic acid, Actigol. Technically, the lumen of the GI tract is outside the body. Enzymes, transporters, etc. prevent entry of unwanted xenobiotics and organisms into the body. For some, it might be easiest to think about the engineering of the GI tract as seen in the diagram. At the oral end, the mouth acts like a chopper and a lubricator. The esophagus is a conduit to the stomach, which serves as a blender, acid sterilizer, and a reservoir. The small intestine is the longest part of the GI tract, extending from the pyloric orifice of the stomach to the ileocecal junction. The small intestine is analogous to a reaction vessel. Bile salts from the gallbladder act as a detergent, enzymes from the pancreas perform digestive functions, and bicarbonate from the pancreas neutralizes GI acid to protect the duodenum. The jejunum is the site of absorption of most nutrients in healthy folks. Its many folds, the folds of Kettering and villi, increase the surface area really markedly. The ileum has fewer folds and shorter villi. It's more closely involved in the reabsorption of conjugated bile acids, but it can serve as a site of absorption if malabsorption occurs at more proximal sites. The colon is a reservoir for the storage of waste prior to the elimination. Generally, there are few active transporters in the colon. The colon is the site of reclamation of many intestinal constituents: fluids, short-chain fatty acids produced by bacterial fermentation, and bile acids and drugs that undergo deconjugation by bacteria. Our colons host an entire ecosystem, the microbiome. The importance of the microbiome to our health is becoming increasingly apparent. This diagram provides a bit of pharmacology-related detail relevant to the GI tract. First, what kind of G-protein coupled receptor could be predicted to cause relaxation of most sphincters? And remember, sphincters are smooth muscle. So it'll be a GS protein coupled receptor. Now, what type of G protein coupled receptor could be predicted to cause contraction of sphincters? Well, remember, GQ lets the calcium in, and calcium makes things happen. Since the GI tract stretches from the mouth to the anus and comprises organs that drain into it, the liver, the gallbladder, the pancreas, local regulation alone is just not quite enough. The enteric nervous system serves to regulate the motility and secretory responses of the gut and to integrate this regulation with info from the CNS. Paracrine and immune messengers act locally to modulate endocrine and neurocrine gut signaling. The neurons of the ENS can be considered to perform functions in 2 primary areas. There are program circuits that receive inputs regarding the physiologic status of the intestine, and they evoke appropriate changes in smooth muscle, mucosa, glands, and blood vessels. There's also integration circuits that relay the info to the CNS and integrate info from the CNS to modify functions. Both the parasympathetic nervous system and the sympathetic nervous system can activate salivation. The parasympathetic nervous system, rest and digest, increases saliva through activation of the GQ-coupled M3 receptor, which is innervated by the facial nerve, cranial nerve 7, the glossopharyngeal nerve, cranial nerve 9. Sympathetic nervous system stimulation of adrenergic beta receptors, the signal originates from T1 to T3 and synapses in the superior cervical ganglia, also increases saliva formation, while when we activate the GI-coupled alpha-2 receptor, we get a decrease in saliva production. Saliva is produced in the oral cavity, and it contains water, ions, glycoproteins, immunoglobulin A, enzymes including lysozyme, salivary lipase, and lingual lipase, growth factors including epidermal growth factor, and some waste products. The pH is normally 6.35 to 6.85. Saliva functions include initial digestion of starches, amylase, lipids, lipase, dilution and buffering, and lubrication. The esophagus penetrates the diaphragm through the esophageal hiatus. The lower esophageal sphincter, LES, relaxes to allow a food bolus to pass into the stomach, then contracts again to prevent esophageal exposure to acid. Generally speaking, signals that increase smooth muscle contraction, like GQ protein-coupled receptor activation, are going to decrease the likelihood that stomach contents will reflux into the esophagus, while those that cause smooth muscle relaxation, GS protein receptor activation, calcium channel block, are going to increase the likelihood of reflux. About 1.5 liters of saliva is produced by the human body every day, and it's essential for carrying out a vital role in lubricating food, digestion, and protecting the oral environment. Saliva is produced by the salivary glands of the body, the parotid, the submandibular, and the sublingual glands. Within the glands, the acinar cells are responsible for the volume of saliva secreted, and the duct cells are responsible for the composition of the saliva. Acinar cells produce initial saliva composed of water, ions, enzymes, and mucus, and it has about the same osmolarity as plasma. The initial saliva then passes into the duct, where the ductal cells absorb sodium and chloride and secrete potassium and bicarb. Salivary bicarb, generated through the actions of the carbonic anhydrase, neutralizes oral bacteria acids, which increase when we feed the bacteria as we eat. Bicarb is therefore very important in maintaining dental health. Final saliva is hypotonic and hypoosmotic compared to plasma, though tonicity changes with flow rate. At high flow rate, ductal cells have less time to act, so saliva is nearly isotonic, while at low flow rate, saliva is more hypotonic. As mentioned, enzymes present in the saliva begin initial digestion, alpha amylase for carbs, lingual lipase for fats. Kallikrein cleaves kininogen into the powerful vasodilator bradykinin. Vasodilation from bradykinin increases salivary blood flow. Acinar cells also secrete IgA. The stomach has three segments: the cardia, the fundus, and the antrum. The surface area of the gastric mucosa is increased by the presence of gastric glands. Different segments of the stomach have different cells and different functions. The glands secrete about 2 liters per day of isotonic fluid. Superficial cells secrete bicarb and mucus that's stimulated by prostaglandin E2. The parietal cells in the corpus secrete acid and intrinsic factor. Importantly, acid is protective against bacteria. Again, acid is protective against bacteria. Chief cells in the corpus and antrum secrete pepsinogens. Pepsins are endopeptidases that initiate protein digestion, and they're activated at a pH of less than 3. We have different kinds of endocrine cells, including the enterochromaffin-like cells in the corpus and antrum that release histamine. And remember, histamine activates acid secretion. G cells in the antrum secrete gastrin, and this stimulates parietal cell expression of HK-ATPase, also known as the proton pump. The D cells in the antrum secrete somatostatin, which inhibits G in parietal cells. It's a sort of check and balance to prevent hyperacidity. Most nutrient absorption occurs in the small intestine. The villi, seen at upper left, increase absorptive surface area. When the acidic chyme enters the small intestine, it's mixed with pancreatic juice and bile. The duodenum secretes cholecystokinin, CCK, to stimulate gallbladder contraction and to relax the hepatopancreatic sphincter. CCK is also a satiety signaling hormone. S cells secrete secretin, which causes pancreatic cells to secrete bicarbonate. The large intestine reabsorbs water, mostly in the transverse colon, and it also stores and eliminates waste. Do you suppose that pancreatic enzyme secretion is stimulated by the PNS or the SNS? Well, if you guessed PNS, you're right, mostly rest and digest. Acinar and duct cells form the functional unit of the exocrine pancreas. Digestive enzymes are stored as inactive zymogens. This inactive state is important to prevent the enzymes from acting on the digestive tract itself. Chymotrypsinogen become chymotrypsin and trypsin to digest proteins and peptides into amino acids. Enterokinase activates the peptidases. Pancreatic lipase digests tri- and monoglycerides into free fatty acids. Pancreatic amylase is an endoenzyme. It doesn't cleave outside bonds, only inner bonds. Alpha-glucosidases break the dipeptide outer bonds. Remember the drug acarbose? It inhibits alpha-glucosidases. Bicarbonate again is secreted by the pancreas, and formation is catalyzed by the enzyme carbonic anhydrase. The return of carbonic anhydrase again. The diagram depicts bicarb formation and secretion. Carbonic anhydrase is important in many organs, including the pancreas, to supply bicarbonate to the duodenum. Carbonic anhydrase converts water and carbon dioxide to form H2CO3, which disassociates into protons and bicarb. The protons are transported into the blood in exchange for sodium, the sodium gradient being maintained by sodium-potassium ATPase. Bicarb is secreted into the intercalated duct in exchange for chloride, and the chloride gradient is maintained by diffusion. Here we're looking at protein digestion. Pancreatic proteolytic enzyme activation begins when trypsinogen is converted to trypsin by enterokinase. Trypsin activates chymotrypsinogen, proelastase, and procarboxypeptidases A and B. Trypsin, chymotrypsin, and elastase are endopeptidases, while carboxypeptidases are exopeptidases that break the outer peptide bonds. Emulsification is a physical process that takes place in the stomach. Phospholipids and proteins facilitate this process. Digestion occurs in the stomach and the duodenum. Solubilization of fats requires bile salts that form mixed micelles. Absorption of fat normally leaves about 5 grams of fat in the stool. Any more is steatorrhea, fatty diarrhea. The sodium chloride-rich fluid secreted by the acinar cells helps to hydrate the dense protein secretions. Fluid secretion is driven by chloride ion. The cystic fibrosis transmembrane conductance regulator protein, CFTR, is an important anion channel that allows for chloride diffusion. In step 1, we see that an inward gradient of sodium is created by sodium-potassium ATPase. In step 2, the sodium-potassium chloride cotransporter causes a net chloride uptake, which is driven by the sodium gradient. Step 3, basolateral potassium channels deliver potassium, which is driven by the accumulation of potassium inside the cell. In step 4, we see that intracellular increases in chloride cause an electrochemical gradient of chloride that causes efflux of chloride into the acinar lumen through apical chloride channels. In step 5, the lumina accumulation causes paracellular shifts of sodium in water and transcellular shifts of water. It's important to note in step 6 that cholecystokinin, CCK, and acetylcholine are potent stimulators of chloride secretion, which leads to water secretion, which increases the volume of the luminal contents, promoting increased liquidity of waste when cholinergic and CCK tone is excessive. The exocrine pancreas is pharmacologically relevant. In pancreatitis, for example, inflammation of the pancreas, pancreatic amylase and lipase are found in the blood diagnostically. Digestion of pancreatic tissue by these enzymes leads to intense abdominal pain radiating to the back, pale fatty stools due to lack of fat digestion, causing steatorrhea. Fatty acids in the stool bind to calcium, which means that calcium can't bind oxalate. Oxalate then gets reabsorbed and filtered to the kidney, where it may precipitate to cause oxalate kidney stones. In chronic pancreatitis, pancreatic hormone replacement is necessary. Cystic fibrosis is a mutation in the CFTR protein, which is responsible again for the secretion of chloride and bicarb. Patients with cystic fibrosis often can't secrete pancreatic enzymes either, so those with CF often need pharmacologic replacement. Importantly also for exam purposes, orlistat is a drug used for weight loss. Its brand name is Ally in the OTC form and Zenacal in the prescription-only form. It inhibits pancreatic lipase, leading to non-digestion of fat, malabsorption of fat, with steatorrhea if the person continues to consume fats. Long-term use can cause fat-soluble vitamin deficiency, loss of A, D, E, and K, and also the risk of oxalate kidney stones. Normally, fatty acids again are absorbed, leaving calcium in the GI tract. The calcium binds to oxalate, causing fecal elimination of oxalate and some calcium. In steatorrhea, the fatty acids bind to the calcium instead, and both get eliminated fecally. Oxalate again is reabsorbed and eliminated through the kidney. The increased renal oxalate can lead to oxalate stones in susceptible patients. Bile acids are also pharmacologically relevant. Bile is secreted by the liver with a pH of about 8, and about a half to 1 liter of bile is secreted by the liver every day. Water, minerals, mucus, bile salts, bilirubin, and cholesterol are all secreted. The bile acids sodium taurocholate and sodium glycocholate emulsify fats in the small intestine to expose a larger surface area of the fat for lipase activity. Emulsification increases absorption of fatty acids and the fat-soluble vitamins A, D, E, and K. Bile acids have a negative feedback for their formation in the liver. They bind to a receptor known as the farnesoid X receptor, FXR. FXR is a bile acid-activated transcription factor that belongs to the nuclear receptor superfamily. When it's activated, it increases expression of the enterokine fibroblast growth factor 19, FGF19, that reduces expression of enzymes needed for the conversion of cholesterol to bile acids, for example CYP7A1. It also increases expression of transporters that pump bile out of the liver, like the bile salt export pump BSEP, and it also inhibits expression of transporters that uptake bile into the liver, for example the sodium taurocholate co-transporting polypeptide NTCP. The drug ursodeoxycholic acid, brand name Actigol, can be used to treat gallstones through this feedback mechanism. Now let's talk briefly about the ENS, the enteric nervous system. It's a very complex reflex circuit. Sensory afferents monitor changes in lumen distension, chemistry, and mechanical changes. When they're activated, they in turn activate interneurons that in turn activate efferent secremotor neurons. The reflex circuitry is modulated by the autonomic nervous system. The parasympathetic nervous system also directly synapses with the ENS through cranial nerve 10, except the distal colon, of course, is innervated by pelvic nerves. Acetylcholine increases contraction of circular muscle at the oral end, while GS receptor activators cause relaxation at the aboral end. At the same time, the longitudinal muscles relax at the oral end and contract at the aboral end, which shortens the distance that the chyme must travel. The motor actions of the GI tract include segmentation, mixing, propulsion, peristalsis, and sphincter contraction that allows for a reservoir function. Let's think about smooth muscle contraction in a pharmacologically relevant fashion. GQ protein coupled receptors and activation of voltage-gated calcium channels are going to cause smooth muscle to contract, while calcium-activated potassium channels are going to cause repolarization. Some signaling players, acetylcholine is going to activate those GQ coupled receptors to cause contraction, while vasoactive intestinal peptide and nitric oxide are going to cause relaxation through their activation of GS coupled receptors. If the lower esophageal sphincter remains in the relaxed state, gastric reflux may lead to heartburn because of the acid splashing up into the esophagus. Rhythmic contractions vary in the fed versus the fasting state in the small intestine. The migrating motor complex of the fasting state is in part mediated by a hormone called motilin. The large intestine has four main functions: fluid and electrolyte resorption, storage, elimination, and short-chain fatty acid absorption, where the short-chain fatty acids arise from bacterial fermentation. The gut-brain axis really does exist. Let's look at satiety and hunger signaling. At the level of the hypothalamus, appetite and satiety regulating neurons, for example those that release pro-opiomelanocortin POMC, a agouti-related peptide or neuropeptide Y, send projections from the arcuate nucleus to the paraventricular nucleus. The anorectic POMC expressing neurons are responsive to systemic signals including leptin, insulin, and metabolites like glucose. Alpha and beta melanocyte stimulating hormone, derived from POMC, mediate the satiety signal through their actions on the melanocortin 4 receptors. Agouti-related peptide neuropeptide Y expressing neurons respond to ghrelin, which is predominantly under the control of the autonomic nervous system. Ghrelin is a hormone released by enteroendocrine cells that act to increase appetite. Ghrelin when your belly's growlin'. Agouti-related peptide antagonizes the action of alpha-melanocyte stimulating hormone at the melanocortin 4 receptor, while the neurotransmitters neuropeptide Y and agouti-related peptide originate an orexigenic or feeding signal through the paraventricular neurons. Satiety is brought about through activation of vagal afferents from the stomach stretch receptors and nutrient-induced release of enteroendocrine factors like cholecystokinin, serotonin, peptide YY, and glucagon-like peptide 1. The vagal afferents send projections to the solitary tract nucleus to bring about meal termination. GLP-1 analogs like liraglutide are used in type 2 diabetes under the brand name Victoza and for weight loss, brand name Saxenda.